Hello, everyone. My name is Diana Cox-Foster. I'm a research leader with USDA ARS Pollinating Insect Research Unit here in Logan, Utah. And today I'd like to speak to you about the efforts and needs of the USDA Agriculture Research Services for native bee monitoring. As some of you may know, within Agriculture Research Service, our scientists are divided up amongst different national programs. Pollinators fall into one national program, NP 305, or crop production. This program has an action plan that focuses on the most critical issues and needs of the U.S. production agriculture within two major areas. The first one is integrated sustainable crop production systems. The second is bees and their pollination. USDA recognizes that all species of bees, honeybees and other species, are crucial for U.S. agriculture and ecosystem health. In addition to honeybees, non-apis bees are effective pollinators of agricultural crops, as well as many native plant species. These native bees perform ecosystem services of value that cannot be estimated. Within Agriculture Research Service, we have three major areas for bee research. The first is bee management, improving bee nutrition and performance. Second is bee health, mitigating the impacts of pathogens, pests, and pesticides. And then lastly, conserving bee diversity and improving bee taxonomy. In addition to these three areas, we have recognized that there are new challenges to bee health. Many of you may have participated in the State of the Science workshops that were held by Elizabeth Hill from the Office of Chief Scientist. You may have also participated in another workshop to help develop a grand challenge project, one that would create synergy amongst all the ARS scientists across the United States that are focused on pollinator health. The title of this project is Creating Pollinator Landscapes and Beekeeping Practices for a Changing Climate, and it's being led by Gloria DeGrande Hoffman of the Carl Hayden Bee Center. Within the Grand Challenge project, there are several objectives. The third objective is one in which we'd like to reach out to you to ask for your help. This objective is to determine the pollinator number, diversity, and the pathogen loads of those bees in plantings of oilseed crops and other types of pollinator plants, and to relate these data back to the nutritional quality and abundance of the plants. The overall goal is to increase plantings that can benefit pollinators and help sustain their populations. Within the project, we hope to evaluate the attractiveness of different plant species based on their floral phenology and the nutritional composition of the plant's pollen. We will assess the diversity of pollinators, their foraging activity amongst the different bee species, and their different pollen resources that they need. We hope to determine the impacts of the plant's nutrition on bee reproduction, overwintering survival, and on the parasite and pathogen levels present in those bees. For these types of surveys, we do need additional scientists from the RCN to help partner with us to make these surveys on different types of crop plants across the United States. For our regular research that we are doing within Agriculture Research Service, I'd like to tell you more about the NP 305, Conserving Bee Diversity, Improving Bee Taxonomy. Within this goal, we hope to improve bee systematics and to develop new tools for rapid bee identification to enhance the understanding of wild bee diversity and to help identify environmental and biological factors that promote wild bee sustainability. For this goal, much of that work is being done here in my research unit in Logan, Utah. We are the USDA ARS Pollinating Insects Biology Management Systematics Research Unit, and we're co-located with Utah State University. Within the unit, we have 6 scientists, including myself, Kelsey Graham, Teresa Pittsinger, Jonathan Koch, Terry Griswold, and Michael Brandstetter. In addition, we're joined by postdocs, very competent technicians, graduate students, and wage payrolls undergraduates. Many thanks to all these people for helping us on this research. We're also the home of the U.S. National Pollinating Insect Collection. This collection began in the 1940s under the guidance of Dr. George Bohart and Frank Parker, with Terry Griswold leading the banner for many years. He has now been joined by Michael Brandstetter, Jonathan Koch, and Kelsey Graham. Major growth in the collection has occurred in the last 10 years. We now have more than 1.8 million specimens with worldwide coverage, but a primary focus on the United States, in particular western parts of the United States. We also have associated with the collection a database that has over 800,000 records and is continuing to be built up. As you're well familiar, each specimen comes with a label, and on that label are very critical, essential data. This data is captured into the database, as well as additional data. And the way that we can link from the specimen into the database is using this matrix code shown on the bottom image on the right. With this matrix code, you can get into the database and learn the metadata as well as additional data. The metadata is very critical and very important. It tells you the details on how and why the specimen was collected. Some of our collections may have had a very specific purpose and may not have been a widespread faunal survey. For example, we've performed many studies of rare plants of concern and looking at their pollination, and we may have only collected and made samples or specimens from bees visiting that plant only and none of the additional plants in the area. We also have specific fields for some of the specimens in there. For many of the species, we have information on their nesting data as well as essential pictures. We may have collected floral hosts and put them into the USU Intermountain Herbarium. We may also have size measurements taken for different species, as well as parasitization data and microsatellite data, including additional fields in there as well. We're also building up other data that can be associated back to our specimens. We may have a fair amount of genomic data, and this is growing rapidly. We have bee genomes that are associated with some of the species that we have in the collection, ultra-conserved element sequences, metabarcoding sequences, and microsatellite data. These are crucial data that can potentially be used to help to identify specimens or species, and to also be used for population genetic studies. We have high density images that are taken with our special Keon's microscope that allow you to zoom in on particular regions to look at morphological characters that may be needed for identifications. At the time that many of the bees were collected, we may have also made pollen slides taken from the pollen that the bees were, the hosts, the floral hosts that the bees were visiting. This allows you to link the bees back to these floral hosts and to create the associations with pollination. We also lastly link publications back to each of our specimens. On the genomic tools that we're hoping to develop, Dr. Michael Brandstetter has been given a major task on this. And he brings with him his tool set in which he has the ability to generate and analyze the ultra-conserved element sequences in bees. These ultra-conserved elements give you sequence data for over 2,000 different genetic loci across the genome of the bees. And these data give you very fine resolution to look at phylogeny and delimit the species. It has many different applications in taxonomy, phylogenetics, and ecology. It's one of Dr. Branstedter's goals to sequence all the US species and global genera. Using UCE data or generating it separately, he's also working on creating a metabarcoding for all the different species in North America. Dr. Branstetter is also involved in the Genome 100 project. This is a brand new project. Congress made available monies this year to create reference quality genome assemblies for over 100 additional bee species. Also involved in leading this project are Jay Evans, Catherine Perrys, and Jean Robinson. With this data, we hope to address questions in bee phylogeny, biology, and health, and may use the data to also find additional population markers for other taxa. We will also gain additional understanding of bee-associated microbes with these bees, help get an insight into the microbiome, as well as pathogens and parasites. In our unit, we've had many previous surveys of native bees. Dr. Terry Griswold has done many of these, primarily in national parks, national monuments, on the lands of the United States Forest Service or Department of Defense. And these have been extensive faunal surveys for the bees. One in particular is found in the western side of California at Pinnacles National Park. Dr. Griswold and his collaborators and students have worked on Pinnacles now for 3 different time points, doing extensive bee surveys across time. We've also made use of the collection to create other databases on surveys of North American bumblebees. Jonathan Koch with Jamie Strange and Terry Griswold helped to create this US Bombas Database. And Jonathan is currently working to expand this and make this more up-to-date on current species distributions of bumblebees in North America. He's also worked with others to create field guides that can be used extensively to identify bumblebees. Current surveys that are ongoing includes one being done by Terry Griswold with students with funding from the BLM. This is to look at the status of the Mojave poppy bee or Perdita meconus, which may be an endangered bee species. The Mojave poppy bee is only found on 2 species of fairly rare poppies, the rare Las Vegas poppy and also the rare dwarf bear claw poppy. This bee is being considered for listing as an endangered species by a US Fish and Wildlife Service. So this data is critical. Another study that we are doing with the survey is here in northern Utah. I'm one of the co-leads on the project. This is to look at the interactions of bee species and their pollen use and their foraging needs. We're basically asking about the nature of the interactions between honeybees and native bees. We're performing one of the first surveys on the native bee species in this mountain area in Utah. And hopefully this data will lead to great insights into the amount of forage that each bee species needs to have reproduction. So with that, I'd like to leave you, ask you to reach out for potential collaboration if you have interests or have a need for doing other surveys. And if you have any questions. With our collection, we're currently not able to have visitors at this time, but we're hoping in the spring of 2022 that we'll be open again and able to have people come in to make use of the collection for research. Thank you for listening.